Hey everybody, welcome to part 3 of this week's topic. Today we're going to talk about one-way analysis of variance, otherwise known as ANOVA. So just to let you know, this material will not be on the final exam. So one-way ANOVA won't be on the final exam, but it's a very important building block for more advanced, more sophisticated statistical modeling, which you will encounter as you go forward in other courses. So let's talk about one-way ANOVA. Sometimes you'll encounter more than one mean. You might have 3 means or many means. How do you compare, for example, 3 different means for 3 different groups? How do we do this? Because before we were only comparing 2 means at a time. You could conduct multiple t-tests. So you could compare group 1 with group 2, group 1 with group 3, group 2 with group 3. So you could do multiple t-tests comparing all these different combinations of means. This seems like a viable solution at first glance, but it does run into problems. So let's revisit this example of light at night. So does light at night make you fat? In this study, 27 mice, they were randomly split into 3 groups. One group had a normal light-dark cycle, which we call LD. One group had bright light on all the time, so we call that the LL group, or light-light, both during the day and at night. And then one group had light during the day and sort of a dim light at night. So you can think of this as sort of a treatment control with another treatment. The outcome for each of these treatment conditions was the amount of body mass gained in grams by the mice. So let's look at this data set again. And this is, again, a real published data set. This is based on real research. This got a lot of press, and here's actually this sort of abstract for the study. So let's look at the data set. We can see that we have 27 cases, 27 rows. Each row is a different mouse. And we can see that we have these 2 variables. We have the outcome variable, the grams gained for each of the mice over the duration of the study period. And then we also have these groups. You have light-light, light-dark, and DM for dim light at night. So let's kind of look at this, what happens when we conduct these sort of comparisons. When we're comparing 2 means at a time, the problem is that we're ignoring the other means. So if you compare the mean of group 1 with group 2, you're ignoring the mean for group 3. So we really want a method that incorporates all the means, all 3 different groups simultaneously. Another problem is that the more means there are to compare, the more groups you have, the more t-tests you need. And this can inflate quite quickly. So when we compare 3 means 2 at a time, meaning we compare group 1 with group 2, group 1 with group 3, group 2 with group 3, you need 3 t-tests. However, if you have 5 groups, so you would have 5 means that you want to compare 2 at a time, you'd need 10 t-tests. And then for a comparison of 10 means 2 at a time, you'd have 45 t-tests. So you can see that the number of tests you actually have to conduct inflates with the number of groups. So there's really this problem with using t-tests to compare group means. So what can we do? Well, we can use this technique called one-way analysis of variance, or ANOVA. It's very commonly used when you have multiple treatment conditions, for example, in this study that we're looking at with fat mice, but it's also used when you have just multiple groups in general. So let's kind of think about what one-way ANOVA is doing. It's used to test the claim that 3 or more population means are equal. So there's that kind of claim in the population. You can think of it as kind of an extension of the 2 independent samples t-test. We call it ANOVA because what it's really doing, the fundamental mechanics of ANOVA, is it's comparing the different sample variances. So just to go over some of the notation or some of the terminology, the categorical variable indicating the number of groups is sometimes called a factor when we talk about one-way ANOVA, and sometimes each group or each category is sometimes called a level. So in the case of the mouse study, we have 1 factor. It's these different treatment control conditions, but we have 3 levels. We have light-light, light-dark, and dim at night, or DM. The null hypothesis for one-way ANOVA is that samples in 2 or more groups are drawn from populations with the same mean values. The alternative is that the samples are not drawn from populations with the same mean values. An additional advantage of ANOVA is that it allows us to partition or sort of separate the variation of the data across groups or levels and those within groups or levels of the factor. This can be very helpful to kind of understand the variability and really understand the sources of variability in your data. So let's look at our data set for mean weight gain. These are a series of box plots, and you can see we have these three conditions. You have the two treatment conditions and the control. And the box plots give you an idea of the differences in terms of variability, but also in terms of the different central tendency for these variables. Now let's think about our orienting questions. Are these groups drawn from the same population, such that the differences among the observations are due entirely to sampling variability, that is, random variation from sample to sample? Or are these groups drawn from different populations so that the differences among the observations are due to differences in mean weight gain across the groups as well as sampling variability? With that, we will continue our discussion of one-way ANOVA in just a moment.